Hello, my name is Jansaya Oralbekova. I'm from Kazakhstan, and this is my country's map. This is my country's flag. Try to remember where you're from. So I went from Wisconsin to Kunshan to get my undergraduate degree. Right now, I'm a junior student of the Kunshan University, which is truly international university in Jiangsu province, Kunshan city. Here, you can get dual degree from US government and from Chinese government. Right now in Dukkenshan University, 16 majors, and one of them is mine. My major is behavioral science with track in psychology. So one of the fundamental courses of this major is biology. So today we will talk about the biology and specifically about the mitosis, which is one of the essential process that is going in our organism. So what is mitosis? Mitosis is a process where a single cell divides into two identical daughter cells. First, during mitosis, one cell divides once to form two identical cells. Second, the major purpose of mitosis is for growth and to replace worn out cells. Third, if not corrected in time, mistakes made during mitosis can result in changes in the DNA that can potentially lead to genetic disorders. So you see how it is essential. Mitosis is divided into 5 different phases, which are interphase, prophase, metaphase, anaphase, and telophase. Here you can see the process of going this mitosis. Then let's start from the interphase. The DNA in the cell is copied in preparation for cell division. This results into identical full steps of chromosomes. Outside of the cell nucleus are two centrosomes, each containing a pair of centrioles. These structures are critical for the process of cell division. During interphase, microtubules extend from these centrosomes. Then goes prophase, where the chromosomes condense into X-shaped structures that can easily seen under a microscope. Each chromosome is composed of two sister chromatids containing identical genetic information. The chromosome pair up so that both copies of chromosome 1 are together, both copies of chromosome 2 are together, and so on. At the end of the prophase, the membrane around the nucleus in the cell dissolves away, releasing the chromosome. The meiotic spindle, consisting of the microtubule and other proteins, extends across the cell between the centrioles as they move to opposite poles of the cell. Then comes the metaphase. The chromosomes line up nearly end to end along the center of the cell. The centrioles are now at opposite poles of the cell with the meiotic spindle fiber extending from them. The meiotic spindle fibers attach to each of the sister chromatids. And then happens anaphase. These sister chromatids are then pulled apart by the meiotic spindle, which pulls one chromatid to one pole and the other chromatid to the opposite pole, as you see in this picture. Then the final stage, at the pole of each cell, a full set of the chromosomes gather together. A membrane forms around each of the chromosome to create a nuclei. The single cell then pinches in the middle to form to separate daughter cells, each containing a full set of chromosomes within a nucleus. This process is known as cytokinesis. So before seeing these phases under the microscope, I prepared warm-up interesting TikTok video. It is my the first time of shooting such kind of videos. Please.
Wait, stop, this doesn't make sense. Hold up, wait, who are you? I am you, what? I am me. No, sir, you are you. Wait, I don't know. 2 is 2. Got 2 bust downs, diamonds blue. 5 minutes.
It was so fun to shoot TikTok video.
Wait, stop,
and it really expresses how mitosis goes.
I am me. No, sir, you are you. Wait, I don't know. 2 is 2. Got 2 bust downs, diamonds blue. 5 minutes.
So let's go to the lab. How we can see it under the microscope. So first of all, let's prepare what we should see, okay? Using a scissors, you should cut a few roots from actively developing onion roots tips and place them in a glass jar with fixated solution, which contains Echelon 321. Then remove 4-5 root tips from a glass watching using forceps. Then cover the root tips with 4-5 drops of chloric acid in the glass watch. Allow 5 minutes for the root tips to soak in the acid. Then use water to gently wash off chloric acid 2-3 times. Then cover the root tips with 3 drops of 0.8% gently in violet stain and let them soak in the stain for 3 minutes. Use a water bottle to wash off excess stain 2-3 times. Then on root one root tip should be placed on a clean glass slide and 1 drop of distilled water should be added to the root tip. Apply pressure to the cover slip with the eraser end of pencil to compress the root tissue. If you want single layered cells, press firmly enough, but be careful not to break the cover slips or the cell walls. So then you can put it in under the microscope and see such pictures and identify which is which phase. Here you can see prophase, metaphase, anaphase, telophase. It is pictures from microscope. So what if mitosis goes wrong? As we previously said, it directly influences to the DNA. Mitosis is an intricate dance precisely choreographed by enzymes and proteins that direct chromosome movement during the cell cycle. If entire chromosomes or segments fail to separate completely, the cell may self-destruct. Generally, errors are harmful, but minor changes in genetic blueprinting may offer an evolutionary edge. The continuance of life depends on balanced cell regulation. Errors in mitosis can disturb the normal regulation of cell growth, rest, and programmed destruction. So, if mitosis goes wrong, some cancer cells can be activated, causing an uncontrolled and irregular replication of cells that form tumors. If tumor suppressor genes are inactivated, cells grow rapidly and irregularly, a condition closely associated with tumorigenesis. It is hard to identify, but at the same time, it is very helpful. Mitosis and wound healing help living organisms rebound from injuries. For instance, active children are prone to sickened knees and elbows. Thanks to mitosis, injuries quickly heal with little no scarring. When skin is scrapped, adjacent cells start multiplying and keep going until the cat is healed over nicely. I think we all had such experience when we were a child. So mitosis help organisms grow, heal wounds, and replace countless cells that are shed every minute. So mitosis is one of the essential process. Thank you all for watching my video. Thank you, my professor and my professors of lab who were with me during this fall 2021 session 2. Bye.